In this video, I'll be explaining why and how we barcode samples for sequencing. So, to begin, if we take a HiSeq 2500, which isn't even the highest capacity machine available, we have about 60 gigabases per lane. And so each lane gives a sequence that we can't really distinguish from others. So it just gives a giant pool of sequence we can, then separate out later perhaps, but that giant pool of sequence is what we have to work with initially. And so if we assume that we want to get 30x coverage of E. coli, so a standard amount for doing genome resequencing, then that gives us capacity for 48 samples per lane. And so if we just sequence one sample per lane, then we'd be wasting an enormous amount of sequence capacity, right? Even in this pretty straightforward case. And so what we do is we use barcodes. They're also called indexes in some cases. So they're short sequence tags that let us to multiplex samples, let us put in those 48 samples in a single lane, and then later on separate out reads that come from each sample and pull them together and treat them separately from the other samples. And so the way we do this is by taking advantage of the Illumina library structure. So if we look at the structure as you've probably gone over before, so we have the P5 and P7 regions at each end. We have the read1 sequencing primer and the read2 sequencing primer and the DNA insert here, which is what we want to sequence. And so there are two basic strategies for barcoding a library like this, and that's inline and adapter. So inline barcodes will be basically part of the DNA insert, so in here or in here essentially. And so those will be read as part of the normal sequencing read, and we'll have to go in later, pull out the barcode as part of that read, and then delete it, of course, from that sequence so that we can get what we actually want to look at. And the other strategy is to use adapter barcodes. So with adapter barcodes, we basically have a separate part of the library structure that's dedicated to the barcode here. And we use a separate primer site here at the P7 region to sequence into this barcode here. And so we basically have an entirely separate read that's associated with this read in a cluster that we use to identify the barcode for any given cluster on a flow cell. And if we're going to design barcodes, there's really a lot of considerations that we have to take advantage of, because as you can think, it's really an issue if a single error can cause us to mistake one barcode for another, right? That's called a barcode bleed, when one barcode is mistaken for another, and we basically have crisscross between samples, and that can be extremely problematic for analysis later on. And so we want to design our barcodes such that it's not that simple, that one error doesn't cause us to mistake it. And so the concept we can take advantage of here is something called Hamming distance. So Hamming distance is the number of positions that with two strings of equal length are different. So in our case, a string is just a sequence, a number of A, G's, C's, and T's in some order. And so if we have two strings, in our case barcodes, and they have to be of equal length, of course, the Hamming distance is the distance between two barcodes, the number of positions at which those two strings are different. It's also which those two barcodes are different. It's also the number of errors we need to mistake one barcode for another. So if we look at this basically cube here, so each vertex is some string, so 0, 1, 0, 1, 1, 0, 1, 1, 1, and each path represents a single mistake that changes us from 0, 1, 0 here to 0, 1, 1 here. So the distance, the number of edges we have to pass through to get from one vertex to another, is the Hamming distance between those vertices, between those strings, or in our case, between those barcodes. So that's what allows us to optimize for Hamming distance to make our barcodes as far apart as possible, so that we can actually distinguish them from each other even if there are errors, or at least we can know that there was an error, right? We won't just automatically see 0, 1, 0 as 0, 0, 1. If we get a single error, we'll see either 0, 1, 1 or 0, 0, 0. Another consideration is base distribution. So you have to remember that Illumina sequencing is just a camera staring at a lot of fluorescent dots on a flow cell. And so we have an issue that if if we're sequencing our barcodes across a flow cell across a lane, and all of them have an A at the same position, it's going to be really hard to tell apart the clusters from each other to see what's where. And so what we want is to have an even distribution of each base at each position, so that we don't have that issue at all. We just see a nice even distribution of bases at each position on the flow cell, and we can tell them apart very easily. And we want to avoid homopolymers, so meaning we don't want to have A in a row, G, whatever, any repeating bases in a row, because what that does is creates a situation where we can't say with as much accuracy, are there 6 A's in a row, 7 A's in a row? It becomes a lot harder to tell that apart, to tell how many there are there. And so we want to avoid that in barcodes. And finally, we want diversity. We want to have at least 4 barcodes per lane, generally speaking, just so that we're not, basically, when we're identifying clusters early on, it's hard to distinguish things from each other if everything looks the same. And so by having 4 barcodes per lane, we make our reads diverse, make our library diverse, and that helps us to separate things out to identify clusters. And finally, there's demultiplexing. So now that we have our reads, we need to separate them into our samples by using the barcodes. And generally, this will be done for you before you get your data from a sequencing facility, but there are cases where you might do this yourself. For example, if you make up custom barcodes, you might do it yourself. And there are a lot of options. Cassava is probably the most common. Something just to look up, you have to do this. We're not really going to go into too much detail about how to do this, I don't think. But you do have a choice here. Do you try to resolve errors or throw them out? So that is, if we have some error in our read of the barcode, and it's clearly not exactly what it should be, do we try to say, well, it's closest to this actual barcode, therefore we're just going to bin it in there? Or do we throw out any errors? So that's something that you have to decide. What is the error rate that you'll tolerate?